spring run Chinook salmon occurred on the San Joaquin River, and Friant Dam went in and essentially cut the population off from its spawning habitat. And after a few years of not being able to spawn and reproduce, the population became locally extinct on the San Joaquin. The San Joaquin River Restoration Program is an effort to reintroduce a naturally reproducing, self-sustaining population of spring run salmon to the San Joaquin River. In order to achieve the restoration goal and meet population objectives, hatchery augmentation of the natural population would be needed. So the Salmon Conservation and Research Facility, or SCARF as we call it, is to raise spring run Chinook salmon for the San Joaquin River Restoration Program, and is a broodstock hatchery program. We collect donor stock from Feather River Hatchery as eggs. They're brought into our facility as juveniles. And then after 3 or 4 years, they're raised to adulthood, to sexual maturity, at which time we spawn them, and then their offspring, production fish, are what we release to the river to support reintroduction.
We use this broad mating matrix that was developed in advance based on genetics. And we scan each fish. Each fish has an RFID tag so that we know who the fish is, what the genetics are. And we sort the fish based upon how related they are to the female. Each female will either be spawned with 2 males or with 4 males, depending on how large the female is, to have a very genetically diverse population.
4480-700.
Once everything is properly sorted, we bring them into our spawning room and we spawn each of the individuals. We use air spawning here to strip the eggs out of the female. Once we do that, we segregate her eggs into 2 or 4 sections, and then each of those sections are spawned with a different male. Those are incubated for about 2 minutes, and then they go off and are disinfected for 30 minutes, and then they are incubated until they hatch here at the facility.